Okay, what I'm going to do now is I like to give this presentation as a background for everything else on beekeeping, because this is about the bee biology, that how the beekeeper can look at the hive and make management decisions based upon what he sees rather than what somebody has told him. So this is a good basis for everything else that I'll be talking about this week. Okay, so there we go. Here's my basic recipe for keeping healthy hives. I'll show this slide again at the end, and it's on my website that you can take a look at any time. And that's what we do. We run young queens, good nutrition, manage varroa as our main parasite, minimize toxins, either agricultural toxins or toxins that you put in the hive to control varroa, or toxic things like that rust fungus spores. And the number one thing is to be proactive rather than reactive. The beekeeper who's successful, he's already gotten everything done and taken care of before the beekeepers who are less successful are even wondering whether they should be doing that particular thing. So try to be ahead of the curve. There's two terms, recruitment and attrition. What you understand is if you were in a war, you can keep fighting that war as long as you can keep recruiting new soldiers. The moment your recruitment falls off, you're going to start losing that war. And the way that bees deal with most problems with pests, with pathogens, with any kind of with pesticides, is simply by recruiting more workers. They just keep producing more workers. As long as they can keep producing healthy young workers, which is dependent largely upon the protein coming into the hive via pollen, they can handle almost everything. The other flip side of recruitment is attrition. And attrition is the bees dying. And they die either from old age or they die from toxicity, die from infection. The honeybee has an instinct hardwired into it not to die within the cavity. So the first thing to think of if you're a beekeeper is you have this vision of this wooden rectangular boxes and the bees in there and your 9 or your 10 frames and all that. And the bees don't share that vision with you at all. They're living in a hollow tree. You have an idea maybe that the bees realize that you exist, and that they don't share that vision. You don't even exist to the beehives. You're an occasional intruder that that invades their tree cavity. If you keep the mindset that as far as the bees are concerned, they're living in a hollow tree as they always have, and you're just an occasional intruder, then you can have a much better idea of understanding what's happening inside in the hive. So just forget that they exist in a hive, because that's not what their behavior is about. The bees rarely will die within the hive. They will always use their last bit of energy to fly out of the hive so that they don't have their sisters don't have to remove them from that tree cavity. If a bee starts getting sick, I should say if a bee starts feeling like it might be getting sick, it will fly out and not come back. That's called altruistic self-removal. And that's one of the ways that bees deal with any kind of infection. So if a bee has a bacterial infection or a virus infection, starts to feel sick, it just flies out before it can transmit that pathogen to its sisters. When that goes into a feedback loop, then you get this CCD, the sudden colony collapse, where a colony just depopulates itself in 3 or 4 days and the bees just disappear very quickly. That's this altruistic self-removal going out of control. But this happens all the time, this attrition of the bees flying off. When I plot out the math on this and look at how many, the daily change in bees right here, you can see during the spring buildup, a good colony will build up at about 500 additional workers per day. So let me give you a little mathematics test right here. If the queen is laying 1,500 eggs a day and the colony is building up at 500 bees per day, how many bees are dying per day? 1,000 bees are dying every single day from old age. Okay? So what I find is with people when I say to do an alcohol wash to look for varroa, and you're going to sacrifice 300 bees, they go, oh my God, sacrifice 300 bees? There's 1,000 bees dying a day, every day in the springtime. During the summer, late summer when you'll be doing it, there's 1,500 to 2,000 bees per day dying in that hive just of old age. the alcohol wash is nothing. It's just a little blip. It's a few hours of a queen's egg laying. So don't get hung up on that sacrificing of a few bees in order to save the colony or to save an operation. You kill 300 bees to save 50,000 bees. So all your management decisions can be put into one of two categories. And what I suggest is before you ever take the lid off a hive, know in advance what your intent is. Know what you're going to be looking for, and know what you're going to do when you see each different thing. So you're not standing there wondering what you're going to do. Understand why you do every single action. And let me get a feeling for this audience. How many of you have been keeping bees for less than 5 years? Okay. How many of you have fewer than 100 hives? Okay. So the problem is for the small-scale beekeeper or the new beekeeper, they really don't know what they're looking for, and they're very inefficient. A professional beekeeper, you do a hive inspection in about 15 seconds or 30 seconds. It's open, it's closed, it's done. The bees hardly even know you've been in the hive. You've learned everything that you need to know for your management decisions, and then you do your management decisions. What I'm going to try to help you to do here is, in this slideshow, is show you how we do this. I do have 3 suggestions, especially for you beginning beekeepers. The first thing is copy somebody in your neighborhood, not somebody from 20 miles away, but somebody who keeps bees right in your area who's consistently successful. Now, let me define consistently successful. Successful doesn't mean you're the loudest beekeeper. It doesn't mean you're the one that's always interviewed by the newspapers. It doesn't mean you're the one who attends all the meetings. It means your main problem is you have too many bees every spring. You can't get rid of your extra bees. So any successful beekeeper, the biggest problem is, in order to keep your colonies from swarming in the spring, you've got to be selling bees because you have so many extra bees on hand. If that beekeeper is needing to purchase bees or is complaining, that is not a successful beekeeper. So that's the easiest way to tell whether somebody's successful or not by how much noise they make, but how many extra bees they have to get rid of every spring. The second thing is, bees are a wonderful animal, and you can have a spiritual experience keeping honeybees. And I'll be the first one to say it's just the incredible thing. But as far as maintaining them, it's animal husbandry. Just like any other animal. Whether you're keeping sheep or goats or rabbits or keeping a cat healthy, they're a little harder to keep care of than keeping a cat or a dog, but they will require a certain amount of investment of your time, and it's just basic animal husbandry. So if you have any background in any kind of animal husbandry, it's all the same type of things apply with bees. And the third thing is, don't ask another beekeeper what to do, and don't look on the internet, for God's sake, what to do with beekeeping. Just shut the internet off, and what I'm going to do is try to help you to learn how to ask the bees. And the way you ask the bees what to do is by reading the combs. What this is I have a series of about 14 articles published in the American Bee Journal, and they're on my website. This is a summation of that series of articles. I want to take you over the course of the year. The place to read the combs is at this interface right here, right between the honey and the brood. This is a dynamic interface within the hive, and this will tell you almost anything you need to know about the condition of that hive and the colony's needs. So let's go over this interface. Excuse me. These are the energy stores. This is where the bees, as an energy storage, what they store is sugar, and they store it in the form of honey. This is like having a reserve of coal or heating oil and food, as far as energy type of food, stored in your house. This is the main protein demand, is the brood rearing the next generation of bees. All that recruitment of bees requires a lot of protein. Bees are made out of protein. A strong hive completely turns its population over in about 42 days. So it means you have to have enough protein to replace every single bee in the hive every 42 days. You have these small interfaces now. This single row of cells right below the honey and above the bee bread is the energy interface. And one of two things will be happening right there. They'll either be uncapping cells and exposing stored honey in order to get energy if there's not enough energy coming in the form of nectar, or if there's nectar coming in, they will be as they remove the bee bread, they will be putting fresh nectar into those cells and curing it into honey for storage. This is the protein interface of the hive. So when the foragers are bringing in pollen, they pack it in the cells, and the nurse bees immediately eat that pollen. It's tough in a beehive to store pollen because there's lots of things that would like to eat it. There's insects, there's bacteria, there's fungi that will readily eat up that pollen. So the first thing the bees want to do is to consume it and put it inside their bodies. That's the safest place. So the nurse bees eat it as fast as the foragers bring it in and digest it and convert it into vitellogenin and store it within their bodies. And they pack their guts full of it, and then they convert that vitellogenin into their fat bodies, and they store it in their abdomen. So the safest place for the bees to store that protein is inside their bodies. Now, if they can't eat it all, then they need to preserve it. And at that point, then, they pack the air out of it, add a little bit of nectar to it, and they allow the lactobacteria to produce lactic acid, do a lactic acid fermentation, and it stores it like you store sauerkraut or pickles. It's not as nutritious as the bees. That's another common misconception on the web. Fresh pollen coming straight in is the most nutritious. It's only stored just for the bees to be able, so that it doesn't rot. The bees can consume it later, but they actually do better on the fresh pollen coming right in. Now, the only bees in the whole hive that have the capacity to digest pollen are the nurse bees. The bees go through 4 stages. They emerge, they're cleaner bees for about the first 3 days. Then they act as nurse bees for about 12 days. Then they act as mid-age bees for an indefinite period of time until, as Eve was saying, when the ethyl oleate from the foragers drops off, then the mid-age bees transition to foragers. The mid-age bees are the general purpose bees within the hive. But the only bees in the hive that have the enzymes to digest pollen are the nurse bees. Once they start shifting to mid-age and foraging, they give up the proteolytic enzymes that digest protein, and they start doing sugar-digesting enzymes. The nurse bees then do the digestion of pollen for the entire hive. They convert it into the currency of protein in the hive, which is the jelly, and the jelly then is passed mouth to mouth to any other bee in the hive. So if there's a forager bee coming back from a foraging flight, and it needs to repair its wing muscles, it needs some protein to do that. It cannot digest pollen itself. Pollen just goes right through their guts and digested. They need to beg a meal of jelly from the nurse bees, and nurse bees give them just enough jelly for them to get that protein input. The queen bee lives on a diet of jelly. It's the perfect food for a bee. Her feces look like water. It's just digested perfectly. And the jelly goes to the larva also. The point is there's a communal stomach within the hive, and this trophylactic exchange where the bees are constantly passing this jelly tells every bee in the hive in real time, all the time, the protein quality of the hive. If the nurses start getting short on incoming pollen, they start cutting back on jelly production, and the foragers immediately know that because when they beg for a jelly meal, the nurses can't give them the full amount, and then the foragers then know, wow, we're getting a little bit short on protein, we better start foraging for pollen. You can see right here where I was doing nosema squashes in a Ziploc bag, that on the left side where I squashed some nurse bees, you can see how the water turns yellow from all the pollen in their guts. On the right side, where I took foragers from the entrance and squashed them, there's no pollen in the guts of the foragers. So it's only the nurse bees that eat the pollen. Now, when that fresh pollen starts coming in, you got to understand this. As Eve said, by the way, I have followed Eve's research for years and years and years. I summarized it in his whole talk on a few graphics that I created under the pheromones in the hive. So if you want to see a summary in graphical form of everything Eve talked about, it's on my website. And my hat's off to Eve for the incredible amount of work and all the researches that he's influenced over the years. Okay, understand this. In the hive, it's pitch black. The foragers have no idea what's happening outside in the environment. The only way they can tell is by the presence of the pollen or the nectar coming in from the field bees. So the foragers, or the nurse bees, always have a really good idea of what's happening out inside the environment by simply walking to the edge of the brood nest and assessing the amount of pollen that is stored right there. If there's extra pollen stored, that means that the foragers are bringing in plenty of pollen, and that tells the nurse bees, gives them a very clear signal that says, outside where you can't see, conditions are really good, lots of pollen coming in, you need to rear brood. You need to feed the queen lots of jelly. You need to feed every single larva. Now, if the pollen starts getting cut back, then the nurse bees don't feed the larvae. They eat the eggs as fast as the queen will lay them. So the queen will lay 1,000 eggs a day, and the nurse bees follow right behind her and eat those eggs right up. Or if it gets a little more restricted, they start eating the larvae. The bees consider the larvae also as a reserve of protein, a safe place to store that protein. So their window to the outside world is how much of that pollen and how much nectar is coming into the hive. Then when there's young brood in the hive, they put off that pheromone. You've talked about E-beta-ocimene. This E-beta-ocimene is the signal that they have every other bee in the hive, we have a viable queen. We have a good ovary. This superorganism, the ovary, which is the queen of the superorganism, is functioning, and we can tell because we can smell the signal from the young larvae. And if they don't smell the young larval signal, they immediately know that something's wrong in the hive. That means that they better either start rearing a replacement queen, a supersedure queen, or replace that queen, or that something, I'll get to that later when we talk about swarming. But that's a clear signal to the bees and the rest of the hive whether or not the queen is functioning, whether or not they are rearing brood by just the odor of E-beta-ocimene. So let's look at the population dynamics. So this is by the way, let me apologize here. Sal, I didn't want to change all the dates, so ignore all the dates. All my graphs will start on the winter solstice on the left and summer solstice in the middle and winter solstice on the right. So just ignore all the dates. I thought about changing them all, and I thought, oh God, that's just too much work. Okay, I did for my Varroa graphs for tomorrow, the next day, whenever I do that. I did change the dates so you guys can actually figure your treatments. So I did this one for over two years. And the point is, like when I was down in Veracruz, Mexico recently, beekeepers have this misconception that the bee's year has to do with the calendar, or has to do with the length of daylight. It has nothing to do with the calendar or length of daylight. It has to do with pollen. So in late summer in Veracruz, Mexico, that's when their winter starts. And in winter, that is when their spring starts. And the reason is, it gets dry down there in late summer. There's no pollen coming in. That's winter to the bees. There's no food. They can't rear brood, so they form winter bees, the diatotinous bees, and they go into their winter clusters to survive the pollen-free period. Wintertime, near the winter solstice, it starts just before the winter solstice. It starts raining, the plants start blooming, pollen starts coming in. That's their springtime. So they start their springtime before the winter solstice, and the shortest days of the year, they're building up, getting ready for the winter honey flow. The point is winter to the bees has nothing to do with day length. It has to do with pollen availability. And you can see here, I did a pollen chart for my area in California with the early spring bloom, the big almond bloom, the dearth after the fruit bloom, and then we have a midsummer bloom, and then it tapers off. And you see that the population of the hive just follows right behind whatever the pollen income is. Now this graph here is, I took data from incredible research project by Lloyd Harris for his master's thesis back in 1976 and 77, where he went out to a group of 30 hives, and every 12 days, the period of time that brood is sealed, if you check every 12 days and you measure the number of cells of sealed brood and divide it by 12, that tells you what? How many eggs the queen lays a day. Okay? If you measure how much sealed brood there is every 12 days and divide the number of cells by 12, you can calculate how many eggs the queen was laying over the past 12 days. So every 12 days he would go out there, he'd measure the amount of sealed brood in the hive, and he would mark 100 newly emerged bees with paint. And then the next 12 days he'd come back, he'd count how many of those newly emerged bees were still alive in the hive and mark another 100 bees with paint. And then 12 days later, do it again for 30 hives for 2 years. Incredible. And then he sat on that data. He published his master's thesis and sat on that data for about 20 years before he started publishing it. So I got a hold of it and started talking to him. I said, hey, can I put your data in a more graphic form? So I created this graph right here. And this is all over my website. And we're going to go through this. So this is from winter solstice to midsummer to winter solstice over here. And when I started looking at this, I could break this down into whatever this is, 12 or 14 biological periods of time where the colony does something differently. So I'll go through each of these very quickly. So we're going to start now. This is from Manitoba, Canada, where they do have a long, severe winter where the bees shut down and there's no pollen for about five months. So we're going to start at winter solstice right here. And they had started a little tiny bit of brood rearing in the middle of winter. I don't know what the stimulus for that is, but you can put bees in a shed, close it off completely from the sun, so it's completely black inside that shed, control the temperature so it's constant all the time. This is very common, shed wintering in Canada, and they're going to still start brood rearing at the right time, independent of any external factor that we could possibly see, and we don't know exactly how they do that. It may just be from certain attrition of the bees or whatever, but they'll often start rearing some bees in midwinter. He actually, one of his colonies, came out of the 5-month winter storage stronger than it went in. They raised more bees during the winter, completely enclosed with no flight, than they had in the more bees, enough bees that the population was larger coming out than it was going in. So they can rear brood during the winter. So on this graph, here's what it shows. The top of the colored is the total colony population in adult bees on this scale, in the thousands over here. The dotted line is the total amount of brood, eggs, larvae, and pupae in the hive at any time. And during the spring, it's not at all unusual to have more bees as brood than you have as adult bees in a hive. Because one adult bee can cover 3 cells of brood. The different colors are the age cohorts. So any bee that is from 0 to 12 days old as an adult is red. 13 to 24 days, 25 to 36 days, 37 to 48 days, and very few bees during the spring or summer ever make it past 48 days. That average longevity is 35 days in that ballpark. And what you'll see here is it changes. We'll get to this later, but when you get these winter bees, these diutinous bees, where they start living longer and they start getting into the blues and stuff. We'll take a look at that. The other thing to look at is I calculated down here along this bottom line the average age of every worker bee in the colony. And you can see we got worker bees that are over 125 days old. His oldest bee he recorded was 300 and some days, oldest worker. Okay, marked bee. They can live for a very long time occasionally. But the average age of the bee in the hive right here is 128 days. You can almost count that as years for a human analogy. So imagine you've got a group of 128-year-old geriatrics inside that hive. And if you look here, it shifts right here. The average age here is 13 days old. And it never gets over 25 days for the rest of the season. So during the spring and summer, you've got a bunch of teenagers and 20-somethings inside the hive. So when you take the lid off, you see a bunch of bees, you think it's the same hive. No, at certain times of the year, you're looking at a bunch of geriatric bees with all the associated problems, and other times you're looking at a bunch of teenagers that they don't have any problems at all. That's why colonies are typically healthy during the spring and summer and have more of a problem when the bees start to age at other times of the year. If you open the hive up, look at the winter cluster right here, pop it open, it looks like it's solid, but actually there is a tight insulating shell of bees around here with all of them poking their heads in because their blood flow goes from the back to the front of their body. So if they have their heads in, they're taking the heat all the time and pushing it back into the hive with their blood flow. And they keep the temperature right at around 55 degrees Fahrenheit, around 10 degrees C. I'll try not to convert too many things because I don't want to make mistakes, but 55 Fahrenheit. And those are the bees that generate the heat for the hive. The bees in the middle don't really have to do much heat generation. And you can tell very easily by just giving me a little puff of smoke right here. Give them a puff of smoke and wait about 10 seconds, and it looks like that. The bees that were in that tight insulating shell, they can't move because they're packed and they're cold. The bees in the center, they were warm and they were not packed, and they'll move away from the smoke. So very easy to see with your own eyes the structure of that winter cluster. And they actually, in the center, they control the environment to the point that the oxygen content drops from about 21% down to about 16%, and the carbon dioxide content goes from a fraction of a percent up to about 6%. It's so tight that they can control the gases inside that winter cluster. And that winter cluster does lose heat. Here's some bees in hives in my yard after a light snowfall, and you can see the heat rises up to the top, And if you're in a cold winter area, it helps the bees a whole lot by putting a little bit of insulation over the top of the hives. So instead of the moisture from that metabolic water condensing on the lid and raining cold drops of water on the cluster during the winter, the insulation allows that moisture to go to the side, drop down, and then the heat of vaporization is recovered back to the hive. When I do the calculations, you can recover about 12% of the energy of the honey that they burn by diverting that warm air down to the side of the hive rather than to the top of the hive. During this period of time, there would be no bee bread in the hive. They've already eaten any stored bee bread up. They're living on their body reserves, and they're able to rear a little bit of brood, but not very much. On their body reserves, they can't rear brood for very long. And what they're waiting for is the first sign from outside that spring is coming. Spring, again, not having anything to do with day length. It's with the first pollen flows. I don't know about here, but northern hemisphere, that is typically tree pollens are the first things that we see. In my area, it's alder tree pollens. And when the first tree pollens come in, you can see them, the bees just immediately start brood rearing. See the brood ramping up right here? This picture from my area, this is alder pollen. Immediately after the winter solstice, brood rearing started up, and this colony's taken off in gear. It's going to start building up. Now here's the problem. Those winter bees, which were long-lived bees, and I'll get to that later on, that can live for 100 days, 200 days, the moment they start rearing brood, they revert to summer bee physiology. And their expected lifespan from that point is about 35 days. So once they start, and it's going to take 20 days for them to rear the first generation of brood, half of them are going to be dead in 35 days. So as your brood rearing goes up, your adult bee population starts dropping from aging. And you can see this happen right here. So look at this. There's almost no mortality of these winter bees for 2 months there. Until when? When do they suddenly start dropping off? Right after they start brood rearing. As soon as they start brood rearing, then all of a sudden those winter bees, which survived just fine for a very long time, start dying very quickly. They're all going to be dead pretty soon, about 2 months after they initiate brood rearing. They'll all be gone. So what you need is, this is called the spring turnover, when they have to rear enough bees, recruit enough bees to take their places before they die of old age. This is the most difficult time in cold winter climates for the bees, is they don't die during the winter. They die during the spring turnover, especially if there's poor weather then. So here's a colony right here. This would be shortly after the winter solstice. This colony, I was grading for almond pollination. They only had 2 frames covered with bees. This big, beautiful brood nest right here. So they're all in. They're putting everything they got into rearing that replacement generation of brood. And the problem is, now you have different pathogens come in. And this is, again, a 2-year graph. And you can see it's different pathogens at different times. During the spring, you have European foul brood and jock brood. You guys don't have European foul brood, do you? Oh, lucky you. It seems to have mutated in the United States over the last decade, and it's become more of a problem than it used to be. It used to just go away by itself, and now it doesn't go away. And then chalk brood. Do you guys have chalk brood? Okay. So those are typically early springtime chilling type diseases. And then after that, during the summer when there's a lot of pollen and you have a bunch of young bees, you don't have much problem. Maybe American foul brood, but almost no other diseases. By late summer, as the colony population is going down and Varroa starts really taking hold, then Varroa and deformed wing virus becomes problems. When it chills during the winter then the paralytic viruses, the acute bee paralysis virus, Cadre bee virus, Israeli acute paralysis virus, they become problems. And then nosema starts at the lowest point during the coolest part of the year. That's when nosema starts to become a problem. And the combination of nosema and a paralytic virus is deadly to a hive. That's where you get this sudden colony collapse. And then it starts back over again, European foul brood and chalk brood. So if you know your seasonality of disease, you know what type of year you need to look for different disease symptoms. This is a photograph I took at 400 magnification. This is a bee's tracheal tube. These striations are its gut tissue. Every one of those little white dots is a nosema spore. That's how thick a nosema is in the gut of a bee infected with Nosema ceranae. They can have close to 1 billion spores on their gut tissue, and Nosema just sucks energy and sucks protein out of the bees. As long as they have a lot of pollen coming in, they can handle that. It doesn't reduce their lifespan much. But if there's a shortage of pollen coming in, then that can be very rough for them. Okay, here's a test for you here. 3-frame colony, little tiny weak one. A month after the winter solstice, cold nights, we get a little bit of frost at night. This was a colony that did not make it to almond pollination. Look at that beautiful patch of brood. Which is greater right now? And no guesses. You'd be prepared to defend your answer. Is recruitment exceeding attrition, or attrition exceeding recruitment? From what you can see. And I don't ever ask for guesses. It's right there in front of your face. You can answer that question definitively. Is recruitment exceeding attrition, or is attrition exceeding recruitment? You're all wrong. Let me ask you one simple question. I already told you, it's after the winter solstice, it's cold, frosty nights. Did that many workers ever thermoregulate that much brood? No, not a chance in hell. Which means the workers are dying faster than they're rearing their brood. Attrition is exceeding recruitment. This colony is on the edge of collapse right now. You might glance at it and go, wow, look at that healthy brood. That colony looks like it's in great shape. All it takes is one frosty night, and that colony will look like that. You can see the band of bee bread out here? Show you over here. There's the bee bread. That's how big that patch was, this colony. And if you look at these bees right here, and you pick them out and take them one at a time and squash them, put them in the scope, 7 out of 10 will have Nosema ceranae infection, sometimes 10 out of 10. And what they did is they had to contract on that frosty night. They abandoned that brood chilled. The next day when they open up, then they'll remove all those chilled larvae, but those bees are dying quickly. You already saw this right here. They're dying quickly because you don't see very many bees on this comb, and one frosty night, and that's it. This is CCD, sudden colony collapse due to Nosema ceranae, and typically a virus with a chilling event at night. But they almost made it. If they didn't have that chilling event, all that brood would have hatched, and that colony would have been out of the woods. They would have been perfectly fine. I see this all the time. And if you look in your colonies at that time of year, you will probably see this also. Other common misconception, they see dysentery in front of the hive. Dysentery has nothing to do directly with Nosema. Nosema ceranae never causes dysentery. Dysentery is from a yeast infection or from undigestible proteins or poor pollen sub or some kind of plant pollen they can't digest. That causes dysentery. Now, if they are having dysentery and they're shitting inside the hive, then that will spread Nosema spores if they already have it. But the dysentery is not from the Nosema. So that's another common misconception. If you see dysentery, that doesn't mean there's Nosema. The only way you can tell if you have Nosema is if you put the bees under the scope and you look and see if you see Nosema spores. People waste, in North America, they waste a ton of money treating with fumagillin for Nosema completely unnecessarily. I had a beekeeper last year, a commercial guy. He had just spent like $10,000 on treatment for Nosema. I said, send me some samples. And I said, dude, you don't have any Nosema at all. Your bees have a yeast infection. That's a whole different thing. The other thing that can happen early in the spring is you saw that stressed colony doing everything they can to keep that brood warm, to keep that the brood fed, and then you go into almonds and you have a gazillion blooms all happen at the same time, and they go out and start foraging. Well, those old bees that are already doing double work they've already lived through the whole winter, and now they're trying to rear all that brood and keep the colony warm, and they go out and start foraging too. They wear themselves out very, very quickly. When a bee starts foraging, it usually only lives for about 8 or 10 more days. And you can have your colonies suddenly collapse right in almonds. And the grower says, wow, what are you bringing me these lousy colonies for? And you can say, they were full of bees just a week ago, and the beekeeper would be right. But it was a bunch of old bees that had not built up and got young bees yet to take their place. So that would be an unsuccessful spring turnover in the almond orchards. Now, once they've done the spring turnover and they've established a brood nest where they have 3 or 4 frames of brood, then they enter this new phase, the linear growth phase. And here's some data from a guy named Nolan back in the 1920s, USDA researcher. And what he shows is it's just a straight linear buildup. There is no curve at all. It's just straight linear buildup for about 60 days, and then the attrition rate of the adult bees comes into equilibrium with the recruitment rate, and the colony then population starts to top out. That top out at maximum population, when you work it out or when I've worked it out mathematically, it's about 42 times the queen's egg laying rate a day. So I'll give you another easy arithmetic problem. The queen's laying 1,000 eggs a day. What's the top out population? It's 42 times the queen's egg laying rate. If the queen's laying 1,000 eggs a day, what's your maximum bee population? 42. Oh, boy, nobody feels really confident on your answers. Yes, 1,000 times 42 is 42,000. Okay, so your colony will reach 42,000. If your queen's laying 1,500 eggs a day, the colony will top out around 60,000 bees. That's where it comes into equilibrium between the queen's egg laying rate and the adult bee survivorship curve. So I made this up this is for my local beekeepers group, where we starting at winter solstice right here, alder first, and I listed the plants right here. Colony builds up, typically swarms right here. Takes 60 to 90 days typically to build up to swarming, then another 30 to 60 days until they build up to full strength for the main honey flow. And this is where we have that blackberry rust I was talking about. There's not much else coming in, especially this part right here. That's when they just pack the combs full of blackberry rust, and it's just death to the. It doesn't kill them; they just dwindle, and they look really lousy. I'll show you how to tell later on. Now, okay, we're going to go back to we're springtime here right now, and we've split all our colonies after almonds. They've built up to singles looking like this, just overflowing with bees. We put the second brood box on. And as long as the weather's good, they're going to just fill that second box full of nectar, and there'll be good, strong colonies. But they're living hand to mouth. No time to ferment that pollen into bee bread. They're eating it as fast as they're coming in. By next morning, there won't be a cell of pollen inside that colony because the nurses will have eaten it all. They're eating it as fast as the foragers can bring it in. The colony's totally healthy. Everything's going just fine as long as nothing changes, like rain. Where I live, I move them up 3,000 feet to 100,000 meters from almonds up to my place, so I take them from spring back to winter. So it starts snowing on them. So they come out of this beautiful almond bloom, and I bring back up to snow country. Okay, and if we get a few days of rain, they've gone through their pollen reserve in a day or two, and it gets pretty rough in the colony. And what they'll do then is they'll start recycling that protein in the eggs. So they'll eat all the eggs, then they'll eat the young larvae, then they eat the older larvae, and the only ones they'll leave will be the capped brood, the pupae, because they don't have to feed them anymore. And what they actually do is on the 5th instar larvae, they'll stop feeding them early, and they'll prematurely cap them. They will still survive to become a worker bee, but they won't be as good a worker bee as they would have been if they had been fed all the way. And that's their only chance for those bees, that colony, to survive, is by that emerging brood that can then, when they emerge, they become a long-lived bee. And I'll talk about that again later. and they just hold on until it stops raining or stops snowing. And this is where I treat my bees better than I treat my own children, because this is our normal bee gear all spring. And people say, Randy, you guys are out feeding bees when it's raining and snowing? I said, well, duh, when it's sunny and warm, they feed themselves. If you're a beekeeper, keeping means when conditions are bad, you keep your bees. This spring, we worked in this gear every single day. Every morning you get up, put your foul weather gear on, and you work your bees all day wearing foul weather gear in order to give them food, because they could not get food from because it was raining too much. We'll also feed them sugar syrup if necessary. We don't feed much sugar syrup. We only feed sugar syrup if we have to, because I hate feeding sugar syrup because it's just a pain in the butt and it's expensive. But this will simulate the nectar flow. So remember, the bees inside where it's dark, they don't know what's happening, but if they got a pollen patty there, then they think that there's plants blooming, and if they got nectar coming in the form of syrup, they think there's a nectar flow, so they will continue their normal spring buildup irrespective of what's happening outside, because we want them to keep building back up so they'll be big for the honey flow. Now, when there's plenty of pollen, then the colony grows very, very rapidly. Again, putting on about 500 bees, additional bees per day. This next 4 slides may be the most important or most useful slides I can show you. People always say, Randy, how can we tell whether the bees need supplemental protein? If you put it on a pollen patty when they don't need it, they'll just suck the sugar out of it and kick all the protein out. You just wasted your money. You just put junk inside the hive. The way you can look is by looking at how much jelly they put around the second instar larvae, the 2-day-old larvae. So not the freshly emerged ones, but the second day. And what you can see, they will start covering the bottom of the cell. Now this colony right here, none of these cells are completely covered to the bottom. They're about 2/3, 3/4 covered with jelly. That means the nurses are holding back a little bit. They're not giving them the full amount of jelly. If this colony was fat with protein, they would cover the bottom of that cell all the way with jelly. These larvae are only getting enough jelly just to survive. So instead of being floated in a pool of jelly and then the larvae eat in a circle, they just keep turning around and around, eating up that jelly, these guys are only fed just enough jelly just to barely survive. They'll live, but they won't be the worker bee that they could have been. How about this colony? You will be, if your colonies look at this, you are an incredible beekeeper. Nothing will go wrong in your hives. You can just pat yourself on the back. God, what a good beekeeper I am. This is telling you this colony will have no problem with disease, no nothing. When you have that much jelly around there, they are in optimal nutritional condition. Here's the colony, here's the eggs. You see the larvae hatching? Barely, little tiny dab, little tiny meals of jelly, not enough to swim around in. So this colony is under nutritional stress. You all got this? Can you see the pictures? That may be the most valuable thing I can show you. This is the first thing we look for every single time we open a hive. We want to do a nutritional assessment of that hive, and that will tell us whether we need to move the bees to a better location, whether they're going to be fine, or whether we need to do supplemental feeding on them. Look at this. Drop in brood rearing right there. Right there. I was curious. Anybody idea? That would be late springtime. Any idea why you have drop in brood rearing? Yeah, that's what I would think. So luckily in this day and age, you can go on the internet and you can read Lloyd's thesis and you can find out the location of the hives and the month and look at weather history, and you can download exactly what the temperature and the weather was for that day back in 1976, which I did, of course. And yeah, a cold rainstorm moved in. Cold rainstorm moved in, the bees couldn't fly, stopped bringing in pollen, and even though they're building up like crazy, they stop immediately. Within hours of when it starts raining, the nurses start cutting back on the amount of jelly that they give to the larvae. Within hours. They've done that experiment. Carl Krailsham did that with sprinklers over hives. This immediate response to lack of pollen coming in. Okay, so now they've built up. We now get to the swarming impulse. The swarming impulse typically happens when the tree cavity starts to fill up with bees and comb, and they've just about topped out on the amount of sealed brood in the hive. You can see the amount of sealed brood is just about to the top level. And then a nectar flow comes in, and they start, this is fresher honey right here, coming in, they start backfilling with fresh nectar, and as these cells of pollen get eaten by the nurse bees, they get filled back in with nectar, and then as this brood emerges, then the pollen gets put here, and the brood nest gets pushed down, and the bees start building more comb beneath. But if they've filled the tree cavity and there's no place to go, all of a sudden, there's no place for the queen to lay any more eggs because they're completely filled up. And now what is missing 3 days later in that hive? E-beta-ocimene. The young larval pheromone. 3 days later, if the queen's not laying eggs, there's no E-beta-ocimene, and the whole colony knows, oh my God, something's wrong with the queen. Plus now they have a big population that's diluted the queen mandibular pheromone, and they go, oh, because it's pitch black inside that tree. But that tells them, wow, we filled the cavity. There's no more room for the queen to lay. We're completely packed full of sealed brood. And at that point, there's only one reason for a colony of bees to be alive, to exist. And that is to do what? To reproduce. And they start building queen cells. So if you're trying to do swarm control, understand what the cues are that trigger queen rearing, that tells the bees that it's time for them to swarm, and then do something to change that. Putting in more combs of sealed brood, putting in foundation. You guys, again, you have this beautiful vision. If I put these frames of foundation in, the bees are going to draw them out, make them into comb and all that. The bees do not share that vision with you. Putting in combs of foundation means nothing to the hive as far as swarm control, because it's not draw and comb, it's not cells. You got to put draw and comb in there. And this is the time to make splits. The proactive beekeeper, which would be us, is already making splits prior to them swarming. So we bring our bees back, we put them in staging yards. Eric goes out there, he just starts popping the lids. If it's wall-to-wall bees, we split that colony up. If it's not wall-to-wall bees, he puts the lid back on, because we know when they hit that wall-to-wall, 20 frames covered with bees, they're going to start making queen cells. So we proactively prevent all of our colonies from swarming by splitting them at that time. Most of them. Now, the colony recovers very quickly after swarming, because when the larvae and pupae are in the cells, they're stacked like cordwood, like this. They take up very little space. When that worker bee emerges, it turns sideways, and now one worker covers 3 cells. So you have a 3-to-1 expansion. 1 full frame of sealed brood emerges to cover how many frames? 3 frames with workers. So in a swarm, you can have 50 to 70% of your bees, adult bees, fly out of the hive, and a week later you don't even notice it, because the bees coming out of the sealed brood, along with their varroa, now repopulates the hives. This also is the most rapid time for varroa buildup. The R value, intrinsic rate of reproduction for varroa, is the highest at this point in time, just at swarming time, when they're rearing a lot of drone brood and they have a very high pupa to bee ratio. Then what we're looking for is the main honey flow. Now, I don't know what your honey flows are around here, but typically you have a spring buildup and then a main honey flow. But some places they don't work in that order. So this is generic here. And ideally, you will, if you had a locally adapted stock of bees that had evolved in your area for many, many, many generations, they would synchronize based upon the plant phenology, reaching their maximum strength at the beginning of the main honey flow. If they get to maximum strength before the main honey flow, they got too many mouths to feed, and they might starve before the honey flow happens. If they reach maximum strength after the main honey flow, then they got to feed a bunch of extra mouths that aren't going to help them get through the winter, and they may starve before winter. So ideally, the most efficient thing for the colony population to do is to reach its maximum strength just prior to the main honey flow. And if you're a beekeeper, the most efficient thing for you to do is to manage your bees so they reach maximum strength just prior to your main honey flow, or to whatever honey flow that you're going to go to. If you're going to go to 2 honey flows in a year, then you're going to build them up to reach maximum strength before each, just at the beginning of the main honey flow. Then the honey flow begins, and you can start shaking nectar out of the combs. This is a shake on blackberry nectar. The year before the drought in California, 5 years ago, taken early in the morning. So, and our honey cures overnight because it's so dry in California. So all that nectar was brought in the morning before 10:30 in the morning. For the next 5 years, we were lucky to get 2 drops of nectar from the same locations with the blackberry at the same stage of bloom. It just, the bees just ignored it, because the plant it bloomed normally, it made fruit normally, but it did not produce nectar. So the main flow starts coming in. They start backfilling nectar, pushing the brood nest down. Now at this point, if you are a bee colony, what's the most important thing for you to do? I'll give you a big hint. They're called honeybees. How's that for a hint? The most important thing to do is to store honey, because the only way you're going to survive the winter if you store a bunch of honey. Do you need to rear any brood at this time of year? No, not at all, because any brood you're rearing now will already, the honey flow will be over by the time those bees could do any good, and there's going to be extra mouths to feed. So what they do is the mid-age bees who process the nectar, instead of taking it up and packing it away, they will start filling the cells within the brood nest and keep the queen from laying. And what you can see during the main honey flow, look at that, the brood ring. Just shut the queen right down. And that's a good thing, because what they want to do is they want to drop their population after the main honey flow. The bees' survival strategy is to build the population up to the main honey flow and then drop that population down and have the minimum number of bees to hold the fort until the next year when they can reproduce again. And then you have these complicated feedback loops using the ethyl oleate pheromone that Eve spoke about and other things that keep this proper ratio of forager bees, mid-age bees, which are your honey processors and your comb builders, and your nurse bees. And I've got several articles on my website that describe how all these feedback loops work. It's just absolutely, it's a miracle, and again, based largely on work that Eve's been involved in. So I thank you again for that. The other thing to keep in mind is the larger the hive, the smaller proportion of workers are involved in brood rearing. And the more proportion of workers will be mid-age or field bees, which are able to either gather or produce honey, store and make beeswax. So ideally, you want your colonies to be as large as possible for making honey production. One large colony will make more honey than two medium-sized colonies, just because it has this better proportion of field bees and mid-age bees. And then what you see is when the nurse bees, now the field bees are not able, it's against the rules in the beehive for them to take their nectar and put it directly into a comb. They got to pass it to a mid-age bee. And that feedback loop, then, by how long it takes a mid-age bee to find a cell to put that nectar in, tells them whether or not they need to build more comb inside the cavity. And when they have trouble finding an empty cell to store it in, that causes them to activate their wax-producing glands, and they start producing beeswax, and you start seeing white wax inside the hive. For you beginners, white wax tells you two really important things. One, those bees need more space right now or they're going to swarm. And two, this is when you draw a foundation. And I get asked all the time by beginning beekeepers, hey, what's the trick for drawing foundation? And I say, there are no tricks. There are no tricks to drawing foundation. The only way to get bees to draw a foundation is if they've filled every single drawn comb full of nectar and have no place else to put it, and then they will start producing wax and they will draw a foundation. No tricks. There's nothing you can pour in the hive, nothing you can do to manipulate. It doesn't make any difference whether you move combs up or down. None of that's all a waste of time. They're only going to draw a foundation if they're making white wax. And then at this point, we trick the bees. So that they don't think that it's time to reproduce, we suddenly enlarge the cavity on them. Here's Tim Ives in Indiana. This is in the middle of the Corn Belt, in the middle of the most GMO neonicotinoid intense area on the planet Earth, producing 350 pounds of honey on there. If you go behind these trees, it's just GMO, neonic, corn, and soybeans as far to the horizon in 360 degrees. I've been up there, visited them, I drove it, I Google mapped it. He just laughs at that. Most of the beekeepers in the Corn Belt just laughed at all the hoopla about all this gloom and doom about the neonics and the GMO crops. Bees can do just fine on them, if not better than they used to. By the way, don't believe everything you read on the internet. I don't know. There's just so much nonsense out there. Okay. Now, after the main honey flow, in most areas you have a dearth where the plants don't produce pollen. Usually you have a lot of spring pollen, then you have fall pollen, but you don't have very much in the summer. If there's not much pollen coming in, then attrition will exceed recruitment, and the bees will start dropping down their population, losing quite a few bees a day, 1,000, 1,500 bees a day, population drop. It can go down pretty fast. So the bees now go into survival mode. There we go. You start dropping down there. And at that point, usually your brood pattern starts to go to hell. It starts getting shot. You don't see very many young larvae. It's not solid because the nurses start eating a lot of the larvae and eating a lot of the eggs rather than raising them. So you get these spotty brood patterns, and you don't see any bee bread stored up here. We've got a little bit of nectar coming in, but there's no bee bread stored there. And at this point, you can create an artificial pollen flow by giving them some pollen sub. And what you got to realize, you're not just feeding them protein, you're changing the age structure of the hive. If they're not doing much recruitment, that means your average age of the bees in the hive is getting old. Older bees tend to be less healthy than younger bees. If you feed them pollen sub at that point, now they start rearing brood, and it shifts the age structure of the hive, and you create a younger hive, a hive full of younger bees. The other flip side, always remember, if you're rearing brood, you're also rearing varroa food. Okay, so you got to do your management accordingly. Know what you're doing. Oh, then introduce our good friend right here. And what happens is, here's the growth curve for the bee population. The black line is the growth curve for the varroa population. Notice it never explodes. And this would be your alcohol wash or your sugar shake, where it looks like the varroa just exploded in the fall. It's not that varroa population increased suddenly, it's that the number of bees carrying that varroa decreased, and it makes the varroa population look much higher. When your varroa population hits about 50 mites per 300 bees, typically viruses go epidemic and the colony starts to crash. The other thing that it does is, here's Lloyd's data again. I overlaid some varroa on it. And there's two things. Here's the alcohol wash coming up. But this is, now instead of showing all the brood, I'm just showing the percentage of pupae in the hive, the number of pupae. And the red is the percent of those pupae that are parasitized by a mite or two. And you notice early in the season, only a tiny proportion of the pupae ever get parasitized by the mite. You get back to this time, now right here, you got 50% of the pupae parasitized by varroa. That colony will not survive. You get about 25% parasitized by varroa, at least in the United States where our viruses have evolved. I don't know, you ask Mark or somebody here what percent your bees can handle. But I'm going to predict when your viruses evolve to take advantage of varroa, you pass 25% of the pupae being infested. How about, Eve, how about in France? Do you have any idea what percent pupae being infested by varroa, your colony collapses at? No, what percent pupae being infested would then result in the colony collapsing? Don't know? Okay. When I plug this into my mite model, you can see how this is alcohol wash count. So a half cup of bees, the mites washed off. So you can see, you don't notice it going up, and then it suddenly looks like it just explodes right here, and you get to that 25% or so of the pupae infested, and your colony will crash. And over the course of the season, in good conditions, you'll go from 100 mites here to 10,000 mites in the hive right there, if you don't do some kind of treatment or something. And I'm going to talk about varroa management and strategies later, but I just thought I'd point out one strategy that is commonly used by many beginning beekeepers, and it's been scientifically proven that wishful thinking has never been an effective strategy for dealing with varroa. It does not work. What happens is when that varroa infestation rate gets up there, varroa starts to become, or viruses become an issue, either the paralytic viruses or typically the deforming virus. So you start getting this epidemics of deforming virus. And we watched deforming virus evolve. We used to see a lot of deformed wing bees. Now we don't see so much. What we typically see is the pupae and the propupae starting to die. Don't see very many bees with deformed wings. They don't make it that far. And then if you are a beginning beekeeper and you want to know why your colony died, I'm going to give you another trick right here. You want to get the sun behind your back. Now, if you look in the sky for the sun, when you find it, you will have blinded yourself and you're not going to see anything. So here's the trick. You look on the ground for the shadow of your head. And when the shadow of your head is directly in front of you, I guarantee the sun will be directly behind your back. Then when you hold the frame right here, you'll have light coming over your shoulders. And you hold the frame horizontally with the top bar away from you, and the light will be shining on the ceilings of the cells. That's this angle right here, and you'll see these white guanine deposits, the fecal deposits from the varroa mite. That tells you your colony died from varroa mites. But before it dies, these mites climb out on these bees. They cover your bees, and they do a behavioral change. Instead of preferring to ride on nurse bees, they shift to older bees, and they hitch rides out to all your neighbors' hives surrounding you. And this mite drift becomes a huge thing. I'll talk about that more another day. Then after fall, if your colony survives to that point, We typically get the fall flows, the fall blooming plants. We get another spike in brood rearing, and this is absolutely critical for going through winter. You want to have these young bees that are raised in the fall free of varroa infestation, which means they're free of viruses. And those young bees, the last ones to emerge, are the ones that are going to become your long-lived bees to make it through the winter. So this would be a rebuilding of the brood in the fall in my area. I should have put in that slide for the rust fungus. I get slides of that brilliant orange, day-glow orange rust fungus in there. But this would be a good pollen right here. So these are the bees that are going to make it, when they emerge, that are going to make it through the winter. Then if you're cold enough that you have a frost, so in Manitoba it happened right here, cold frost kills all the flowers, nothing flowering. The bees immediately respond with queen rearing or brood rearing going to 0. And then the bees do something else. And the best we can figure out so far is that when these last generation of bees emerge, they take a sniff and they smell the queen bee, but they don't smell any E beta ocimene, which means there's no young larvae in the hive. If they don't smell any E beta ocimene, no young larvae, that tells them a very clear message. There's no pollen coming in, so this colony is in survival mode. So the moment they emerge, they know that they're in survival mode, and instead of becoming a nurse bee, they become a dilutinous bee, a long-lived bee. They just pack their body full of, here's a graphic for it, whether they smell the brood pheromone or not, whether they become a nurse bee, a short-lived nurse bee, or a long-lived winter bee. And what they do is they pack their abdomens full of their fat bodies full of vitellogenin, their hypopharyngeal glands, and they go into the winter as fat as they can to survive. And when I've gone out to my hives then, this would be in fall, where normally only a small portion of the bees have pollen in their guts. Every single bee in the hive has a gut full of pollen. They just load up with as much protein as they can. They load their fat bodies, they load their hypopharyngeal glands, they load their guts with as much protein as they can, and then they put it as much in the cells as they can because that's their only protein source for the season until the plants start blooming again. Then you have the fall turnover. In the fall turnover, see this big drop in population right here? All those short-lived summer bees, which only live about 35 days, they fly out of the hive normally to die, leaving behind just the old bees. This is a study I did, started 199 hives. This is their strength going into, so let's see, this was October, December, January. So this is late fall, mid-winter, and then shortly after the winter solstice. These are the number of colonies that had 18 frames of bees, 17 frames of bees, 16 frames of bees. These are the weaker ones, only 7 frames of bees. And this is, so the blue line is the fall strength, the light blue line is the midwinter strength, and then the green line would be their very late winter strength, just before almond pollination. And what you can see, the strong colonies, they lose half their adult population, okay, going into winter. Those bees are all old bees. They're never going to make it through the winter. They just fly off to die. They don't want to die in the cavity. So that's normal. And then what you start having is, these are different viruses. This is deformed wing virus, which usually decreases during winter because you don't have much mite reproduction. But the paralytic viruses start to build up typically midwinter. And again, they're a problem with nosema. This is some data I had. I tracked 36 hives every month. I did nosema spore counts and then crushed 10 bees from each hive and counted what percentage of them were infected with nosema. And it followed the same trend. And this is the inverse of temperature. So the colder it is, the higher the red line. And what you can see is the colder it gets, the more nosema. When it starts to warm back up, nosema starts to disappear, nosema ceranae. So people talk about it being a warm weather nosema. It's a cold weather nosema. It will increase if there's a big pollen flow coming in, because when there's a lot of pollen coming in, nosema increases in the bees' guts, but it doesn't tend to hurt the bees at that time because they have so much nutrition. They live about the same amount of time. Then we're back to midwinter. initiation of brood rearing. So we've gotten all these old winter bees right here now. So there's no bees in the hive hardly at all there that are less than 70 days old right there. They'll start doing a little bit of brood rearing. And at this point, those of us going to almond pollination, we can, if the weather is clear after the winter solstice and the bees are foraging, I don't have to do a thing. If it's rainy and snowy, then we go out there and we put pollen sub in, and as far as the bees are concerned, there's a pollen flow on, and they start building up, so there'll be strong colonies going to almond pollination. We have to take bees, any of you in here pollinate almonds? Okay, almond pollination, it's wintertime, and I move my bees often out of snow to go down and pollinate the almonds, so I have to simulate a pollen flow. Here's my buddy Keith Garrett. This is taken shortly after the winter solstice, where he has colonies so full of bees that he shakes bees and he sells tens of thousands of dollars worth of shook bees to beekeepers who have weak colonies because nobody else can keep their bees strong during the winter. He just puts on a ton of pollen sub. He may put 15 pounds of pollen sub on during the winter on a colony and then shake hundreds and hundreds of dollars of bees out of that colony for sale. He just turns pollen sub into bees. Okay, first tree pollen start coming in. Brood rearing takes off again. There's the alder pollen coming in. Cycle starts over again. Recipe for healthy hives. And I think, there's my website right there. If you go to, let me see if no I don't. There's a short version of this in the beginner's pages, this first year beekeeping I'm working on. I don't have anywhere near enough time to keep up with the website. One of these days, my sons are taking over the operation. I just turned 66. I'm hoping to spend more time getting my website organized, but I'm doing the best I can right now. So how much time do we have? Yeah. No, is there 10 minutes? Okay. You got any questions? I'm happy with questions. Yeah. And remember, you're speaking to them, not to me. Sure, that would be great if you could walk up to the mic. That would help a lot.
Hello, Randy. My question is with regards to the composition of the contents and the size of the venom sac of an old overwintered worker bee. I often find when the beekeeper is stung in spring, that sting can be quite painful, and is more likely to have an allergic reaction.
Absolutely.
So
and I think it has nothing to do with the venom.
is it to do with the composition of the venom?
My guess is it has to do with you weren't stung during the winter.
Right.
I can guarantee you right now, if you get stung all winter long, that doesn't happen.
Yeah.
But if you take a break from getting stung, it will seem more painful and you will typically get more reaction.
Yeah. So is the venom sac increased in size over that period?
I have no idea, but my guess is the explanation for your observation is that you took a break from getting stung during the winter. Probably not that it had anything to do with the venom sac.
Right.
That's my guess. That would be Occam's razor, the most parsimonious explanation. What do you think? He doesn't know either. Sound like a good guess? Okay. Yeah.
Randy,
But it's very common, your first few stings of the year, if you take a break during the winter, to get more reaction for about the first dozen stings. If you work bees all winter long, that does not happen. Your reaction doesn't change. And the stings feel the same. Yeah.
you talked about swarming and the reasons for. Every now and again we get a hive that's got plenty of room. It's got a nice expanding hive, but it suddenly starts building swarm cells.
Yeah.
Why?
We have a name for that. It's called frustration. That happened this year. We because of we had a little bit of pollen flow. The bees started building up big. They all started making swarm cells, and then it rained for a whole month straight. So what they do then is they tear down all their swarm cells because they won't swarm if they don't have good nutrition. And they stopped swarming. I thought, wow, we're out of the woods. They're not going to swarm this year. That's wonderful. So we went ahead and we split the colonies afterwards in the spring. We got them all worked out in singles, so we got them supered up with drawn comb. They got all the room in the world. The main honey flow comes on. By that time, we normally have 0 swarming, and they all decided to swarm at that time. Plenty of room. So yeah, it's just absolutely frustrating. I wish I could, if I could figure out how to stop that from happening, it just totally surprise us. I don't know why.
Randy, one thing I have noticed over the past few years is I always get 1 or 2 hives where the queen stops laying at this time of year.
Uh-huh.
What's the explanation for that?
Okay, I gotta think about what time of year. You're in the middle of winter right now. Well, that's a normal thing in many areas for the queen. This climate is kind of similar to how mine used to be 30 years ago before climate change, and all the queens have stopped laying. They have what? What queens? Oh. Okay.
Most of the colonies have got viable queens in them.
Yeah.
There will be just 1 or 2 that actually do stop. So
Yeah.
1 or 2 colonies have brood breaks, which are obviously an advantage for Varroa.
Right.
I was
But
just wondering...
That's not what you're saying, just a brood break?
No, they just stop laying. It's just something
Wait,
they
what's
do.
the difference between a brood break and stopping laying?
I
They
don't
stop
know.
laying for good.
Well, they just stop laying and then they start laying again.
That's called a brood break.
But that's just something that they do spontaneously, whereas a colony next to it, or another 5 colonies in the same apiary, don't do that.
Oh yeah, with the genetic diversity of bees, if you graft 30 daughters, and I've done this in a lot of yards, you graft like 3 dozen daughter queens, and you manage them exactly the same in their nukes, and you put them in the yard, you got a bell-shaped curve of behaviors, and you're going to have erratic ones that don't build up hardly at all. You're going to have ones that make a ton of honey, and most will be in the middle. You're going to have some that have one behavior and some another. There's that diversity built into the bees. It's not like they're not clones. A bee colony, if a queen mates with 20 drones, you got 20 different patrilines of sisters, each with a different father in that hive, and each one is a team. So imagine a rugby team. I don't know how many players in a rugby team. Imagine there's 20 players in a rugby team. Every hive has 20 different players, and they're going to have different kinds of skills, and every team is going to be different, as opposed to getting a clonal type chicken or a corn plant where they all behave the same. Bees don't, unless you have really, really inbred lines, they don't all behave the same. So that doesn't surprise me at all. We see that kind of variation, and that's the kind of variation that you breed from. If you have a variation in a stock, then you can do a selective breeding program. That's what we do with Varroa. We look for that variation, and you breed from it.
Randy, I was interested in your mention of Nosema. Squashing bees, it seems that if you see Nosema, you just assume that it's Nosema serrani. Do you not have Nosema apis in California?
It largely disappeared. If you looked under the scope enough at Nosema, it's obvious the difference to your eye, whether it's apis or serrani. Nosema apis has a spore that's shaped kind of like this, kind of oval and kind of squarish at the end. Nosema serrani is more tapered at the end, more banana-type shape, and it stands out very, very easily. In the U.S., it's rare. I remember when we were in the middle of it, I visited Australia, West Australia, and I go, wow, this Nosema sure looks different. You guys got Nosema apis, and they told me yes, PCR confirmed it. And then I was up in the state of Oregon, and they had a hobby day, and they had some bees under a microscope showing Nosema, and I said, oh, I'll take a look, and I go, this is Nosema apis. Why do you have Nosema apis? They go, well, we didn't have any fresh bees, so we went to the lab, we took some old specimens that were in alcohol forever, we squashed them. Of course, you were the only one who noticed it, Randy, sorry. No, it's if you've looked at Nosema enough, you can see the difference of the spores. So I don't know what the status is in here, but in the U.S., there's very little Nosema apis anymore.
Yeah, we do PCR diagnostics for Nosemas. We see both of them.
You do?
So I was curious that your definition of CCD is Nosema serrani and an RNA virus.
It's Nosema, either Nosema and a virus together. It can be bad. And even long before we talked about CCD, if you look at the the old literature, the symptom of Nosema apis, if you see a sudden collapse of a colony, that's a symptom of Nosema apis then, and now it just transferred over to Nosema ceranae. As far as I can tell, there's not that much difference between the two as far as the effect on the colony, other than that Nosema apis typically goes away in the spring. It doesn't show up to the fall. Nosema ceranae can overlap and pop up during the summer when you have big pollen flows.
Well, perhaps Mark and James Sainsbury can comment, but we think a more prognostic signature is actually seeing both Nosema ceranae and apis together from a sample.
Wouldn't be surprised. Could be. They both have two different strategies in the bee gut. Nosema apis continues to grow in the gut. Nosema ceranae does a really rapid colonization, just takes over the gut, and then stops making vegetative spores, which are the ones that reproduce in the gut, and shifts to environmental spores, whereas Nosema apis continues to do the vegetative spores, usually at a lower level. So the combination of the two could be bad. And my guess is after your invasion wave gets used to it, somebody, like during the CCD, somebody said, Randy, is there anybody breeding for bees resistant to CCD? And I said, well, as far as I know, I don't know any queen producer who breeds off of dead colonies. So I guess the answer would be yes. So if you just keep breeding off your healthiest colony, Mark and I were chatting here after he spoke this morning, and he asked me about sterilizing combs and stuff like that. And I said, that is one approach, Mark. I said, my approach is I take all my dead out combs and I put them right on my fresh mated out nucleus colonies, and I said, if you can't handle dead out combs, I don't want your genes. Okay? We breed for bees that are tough. I don't want to breed for bees that I got a baby. Florist Nightingale is not my model of beekeeping, okay? I want bees that I can just throw whatever on and they just take care of themselves. Okay, so if you like being Florist Nightingale, God, do all this nursing of your bees and stuff, and sterilization comb and stuff. If you want to be Arnold Schwarzenegger, just put that crap back on the hives, and if they don't make it, don't breed from them. Okay?
Hi. About the nosema and the dysentery symptom of nosema,
Yeah.
it is in the colony loss survey by Colos that dysentery is a symptom, or maybe I'm wrong, but anyway,
It
I've
is in
heard
many
that.
of
And
the
then I read
surveys.
that apis and ceranae, one does have dysentery, the other causes dysentery, and the other one doesn't. I was wondering if you have any history of what is going on with this dysentery and the
What I
two
would
kinds of
say is I will
nosema.
challenge you anywhere in the scientific literature to find any reference ever that anybody has ever associated either form of nosema with dysentery.
So where did the idea come from?
Beekeepers. Yeah. No, I have searched for any reference, even with Nosema apis, and no, it's not there. It's just it's a rumor or a legend that got started. What we call it's a process called reification, which means if somebody says something and you repeat it enough times, it becomes fact. That's called reification. There's a lot of that in the bee literature, like that it takes 21 days from egg to adult. It takes 20 days. It doesn't take 21 days. All the books say 21. It's not a hard thing to test, but there's lots of errors in the bee books.
You haven't mentioned much about drone comb, and I'm just wondering if you do taking off drone comb or taking it away, and what would be the best time of year to do that to reduce your mite population?
You're not going to reduce, I'll talk about that when I talk about varroa, but you don't reduce the mite population by very much, maybe about 14% with each drone comb removal. We put drone combs in all of our hives for that, with the intent of doing mite reduction. And then we just left them in there, and we didn't see that it made a whole lot of difference whether you had drone combs in there or not. But then what we did find is if you have a drone comb in your hive, the bees don't rework worker comb into drone brood, and they don't build drone comb in between the two boxes. Our business, we make as much money selling nukes close to it as we do in almond pollination. Our buyers love to get full, beautiful wall-to-wall frames of worker brood with no patches of drone brood. Well, the reason we run drone combs in every hive right now is we run one drone comb, all of our foundation gets drawn out into beautiful worker brood, and it's a more saleable product. We manage where the bees put the drone comb. You're not going to ever talk the bees out of producing drone comb. So you might as well put it give them a comb to put it in, and then you manage where it is. Each spring after almonds, we cut out that drone comb of all the colonies other than the breeders. Any colonies that we want for breeding, we want them to produce as much drones as possible, and the other ones we cut the drone comb out of. Oh, one more here.
I've been doing it a few years, and I've noticed, like, we'll get one site that would be swarming all the time. We'll be chasing them down, up and down, and we're like, what? We'll change the frames and stuff, and try and give them room, and they still swarm the same place. Is it because there's a heap of pollen and stuff, and they're just going nuts there, or what?
You know, not seeing the site, I couldn't tell you. That would be my guess. I mean, colonies that build up fast, I mean, trying to prevent swarming is like trying to prevent teenagers from having sex. I mean, you can really work at it a whole lot if you want, but you're probably not going to be successful, okay? So, you know, bees would not exist if they didn't reproduce. And you can try to breed against swarming. And what we found is if we just split all of our hives prior to swarming, and I'll show when I do the Varroa thing, I'll show you how to do a split and using oxalic at the same time, you can make your hives almost mite-free, requeen your hives, and prevent swarming all in one easy way of doing it.
Because we had split it, but yeah, no, didn't spray it with.
Yeah, so splitting is the easiest way
Yeah.
to reduce swarming to the minimum level. Most years we can reduce swarming to almost nothing by.
It's just a small site. We had like 7 hives there, and I think we ended up going and getting like 17 swarms from there. You know, it was just like, ah.
Yeah, one of the problems if you're starting off with the bees, you never have enough drawn comb on hand.
Yeah,
drawn
true.
comb is your most valuable tool in beekeeping. You'll make way more honey. You'll be able to reduce swarming. You'll be able to make your colonies build up faster because the bottom box you can put drawn comb in. We never put foundation in a bottom box. It's nothing but drawn comb. Your beekeeping success starts to improve greatly once you have a reservoir of drawn comb to work with.
You can't buy it, unfortunately.
Yeah, I know. It's as long as you're expanding, you're not going to have that. Okay, I guess our time is up, guy. Oh, one last question.
I was wondering about breeding for Varroa resistance in bees. Do you think it's an achievable goal?
Oh, absolutely.
Treatment-free resistance?
Yeah, but the way you go about, I'm going to do a program. One of the workshops will be on that, showing you what we're doing and what I feel is the most reasonable thing to do. Thank you. Thanks.